Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gill Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gill@eapen.info. For listeners familiar with the Scientific Sense podcast, today's format is a bit different. We have 3 experts rather than 1 guest, which has been the case for the last 59 episodes. We're also not discussing specific research papers, but rather hope to accumulate the wisdom of the researchers over the years. We're going to focus on pandemics in general and COVID more specifically. And as usual, we will try to keep this apolitical, but we will look into the impacts of policy and discuss how we could improve as we encounter future pandemics. Welcome, Jeff, Fred, and Jim. We will do a quick round of introductions. Jim, would you like to start?
Sure. Thanks, Gill. My name is Jim Bashkin. I'm a professor of chemistry and biochemistry at the University of Missouri, St. Louis. And my career spans both industry and academia. I spent 12 years at large chemical and pharmaceutical companies, and the rest of my career in academics. And I also co-started my own small pharmaceutical company working on antiviral drug discovery and development.
Great. Fred?
Hi, I'm Frederick Inglis. I'm an assistant professor at the University of Missouri, St. Louis. My educational background's in biology. I'm particularly interested in evolutionary biology. That includes kind of evolving infectious diseases. I mostly work on bacteria, but have some interest in viruses as well.
Great. Jeff?
Yeah, hi, my name is Jeff Smith. I'm a biologist. I study the ecology and evolution of microorganisms, and I'm about to start in Fred's lab at University of Missouri, St. Louis.
Great. Yeah, so I want to start with something fundamental. So the SARS coronavirus 2, COVID, it's an RNA virus. There are differing opinions as to if an RNA-based entity can be considered live, as they cannot replicate by themselves. Is this really a semantic question, or is there something more to it?
So I've thought a lot about this. And in my experience, among the colleagues that I've spoken with this about, I have yet to meet a microbiologist who thinks that viruses are not alive. They're made of the same things that other cellular organisms are. They're made out of proteins and nucleic acids. They have the same kinds of genes. They work the same kind of ways, and they evolve in the same kinds of ways. So I don't think as biologists we have a whole lot to gain or any insight to gain by separating them out from the rest of biology. It's true that they do require cellular organisms to reproduce, but all organisms are dependent on other organisms to reproduce, except for maybe some photosynthetic microbes.
Yeah.
So I don't see that the fact that they eat things from the inside rather than the outside as being fundamental in any particular kind of way.
Okay. Yeah, without knowing a lot about that, Jeff, I was thinking, are there any sort of analogous things that we see between an RNA-based entity and cancer? Jim, I know that you have done a lot of work in this area, and they seem to replicate very fast. And is there any similarity there, Jim?
Well, there could be, and there certainly can be mutations involved in, there are mutations involved in cancers. Viruses can mutate rapidly. They don't all mutate as rapidly as each other. I mean, some viruses mutate more rapidly than others, but some viruses cause cancer. So there's definitely crossover. For example, human papillomavirus, that's only one of a number of small DNA tumor viruses. And so there are definitely crossover properties between viruses and cancer. Some viruses integrate into human DNA, including RNA viruses like HIV, and this can lead to cancers for a variety of reasons, having to do with the fact that human DNA is disrupted, as well as the fact that the immune system is completely disrupted, or not completely, but largely disrupted in the case of AIDS. And so there are definitely crossover properties. I don't know if I can say similarities, but exactly, but areas of common behavior, perhaps.
Common behavior, and especially mutations. So Jeff, do you want to set the stage for what we know about COVID and there's sort of progression and mutation? Are there multiple mutations they're already fighting? Do we expect this to mutate a lot faster? What the implications are?
Well, I can tell you what I know about how COVID arrived in human populations and how we believe it's likely to evolve in human populations.
Yeah.
So COVID is a coronavirus, and coronaviruses in general, the diversity of coronaviruses is in general hosted by bats, but other animals also pick up coronaviruses. Humans have had coronaviruses in the past through SARS in 2003. There was the Middle Eastern Respiratory Syndrome a while back, and there are a couple of coronaviruses already that cause the common cold. This coronavirus is distinct from those, but still part of the same family. In general, they seem to get into human populations not necessarily directly from bats, but often through an intermediate animal, like palm civets for SARS, I think it was, and the camels in the case of the MERS virus. It's still unclear how the specifics of how COVID-19, the virus, SARS-CoV-2, the virus that causes COVID-19, how that arrived in human populations. There are similar viruses in horseshoe bats and in pangolins, but so far none of them are an exact match. So it's possible that there is some other intermediate in there as well.
Sort of intermediate host.
Yes.
So Fred, if you look at infectious diseases more broadly, what do we know? What's the current understanding of the evolution and emergence of infectious diseases worldwide?
Yeah, I think the kind of one of the big driving concepts when people think about how diseases evolve is probably this transmission virulence trade-off hypothesis. So this is an idea that you want to maximize. Evolution is acting to maximize the spread of an infectious agent. That's good. If you're an infectious virus, bacteria, whatever, you want to get to new hosts. And so there's many things that are important there. You don't want to kill the host too quickly. because if you kill off all the hosts, then you can't spread. But often to spread, it requires some kind of damage to the host. Diarrheal diseases causes diarrhea, and that can have devastating effects. Viral diseases require you to, many of them respiratory, require you to cough or sneeze to spread the virus.
Yeah.
It's better if you kind of like lies up a bunch of lung tissue and people are coughing a lot, right? But if you kill too many people, you can't spread. So I think a classic example that's a very interesting one actually is Mixomatosis. So this is the disease-causing agent in, rabbits. And it's really effective. In fact, it kills almost 99% of rabbits it infects. And so it was released, presumably by some Australian farmers, like in the 1900s in Australia, to kill off this kind of devastating plague of rabbits. And at first, tremendous. It killed off all these rabbits. But over time, they discovered that actually Mixomatosis evolved lower virulence, so stopped killing as many rabbits because it couldn't spread. So these kind of hypervirulent strains would go extinct. So that's interesting when you have these kind of jumps from kind of bats or some intermediate kind of host to humans. Is the first stage is a bit dicey. We don't know what's going to happen. It could be that virulence could increase. It could be that it decreases. But usually there's some kind of selection for your transmission and your virulence. Those are the kind of, the two things that something's happening with.
So that's very interesting. So if you take some kind of a ratio, this could be conceptual, but some kind of a ratio, transmission over virulence, you would see sort of, if you optimize that from the virus's perspective or the infectious entity's perspective, you would see some sort of a inverted U-shaped curve, right? It want to have sort of an optimum point between transmission and virulence, so that there's just enough time to jump to the next host, and so this is an evolutionary trait that so over time, by random mutations, they tend to get to that optimum point. Or what do we see? What happens?
Yeah, absolutely. So I mean, it can be that you're so virulent you go extinct. Yeah. So you see you might pop into the population, you kill everybody, you can't spread, and so then you go extinct in that population. But if there's enough time, then you won't. Often what we see is you see very virulent pathogens that become less virulent over time. I think that's the easiest one to observe because they did kill a lot of whatever their host organism is, and then they've stopped or they've reduced the amount of killing because there's been the hypervirulent strains have gone extinct. But equally one can expect the opposite. If you transmit to a new host and there is a lot of contact, there's high population density, for example, maybe you're in a city and there's humans, then in principle one could increase, you can see selection for increased virulence. So both things can happen. It just depends on a lot of the life history traits or how the kind of infectious agent spreads. Mm-hmm. So some of the kind of most deadly diseases that we think of, so you can think of Bacillus anthracis, causative, we think of anthrax. This has a crazy life history. So what happens is it infects ungulates or cows, and then it usually kills them. It produces loads of spores, so tens of billions of spores inside the cow. The cow dies. They can live in the dirt, the topsoil, for hundreds of years, presumably, until another cow comes and feeds on it, and then they kill that cow and they do it again. That's why they can, because of the way that they work, their biology, they can survive. They can cause these like very virulent outbreaks of disease.
Yeah, I hate to be cynical about this. So in this case, it seems like the younger population who are moving around have asymptomatic disease, and the older population who are sort of protected, not moving around, actually have severe disease. So it seems like the virus has figured this out.
Yeah, maybe. I mean, it's possible, right? So it doesn't seem the health consequences in younger people don't seem to be as bad as in older people. That's probably true of many diseases.
It's true for influenza, for example.
Yeah. So
the health consequences can still be severe in young people. It's just that they're not severe nearly as frequently in young people as they are in older people.
Yeah. So one criticism that's out there, Jim, is that there's so many people having differing opinions, even among scientists and professionals, let alone politicians. The public has great difficulty figuring out who to believe and how to go about it. What is your perspective on that? I mean, this is not just for COVID, but more generally, how will we handle this in the future?
Well, I think that we need to have a national policy, and that national policy needs to be divorced from politics. It needs to be based on science and public health considerations alone.
Yeah.
And other countries have managed to do this fairly successfully. For example, the countries in Europe. And they've been relatively nimble in reacting when they found that their policies are starting to fray. For example, Europe has just canceled vacations because they've seen that they opened society up too much, and the number of COVID cases started to increase again. And and this has been enacted over a wide number of countries, and quarantines have been put in place, and borders have been closed, and this didn't take a lot of hand-wringing or months of arguing back and forth. People think of the European Union as an unwieldy organization, but the individual countries acted on their own and did what they needed to do to protect the population.
Yeah, I wonder though, Jim, is there a scale issue? So if you look at the three countries, 1, 2, and 3, in terms of, let's argue, probably badly handled from a policy perspective, from a national policy perspective, that's the US, Brazil, and India. They are large countries with large populations. They have sort of autonomous regions within that country. And so, I wondered that there are two issues there. One is one of scale. It's easy for Denmark or Finland, to have a national policy and implement it effectively. But when you get to some large number, it becomes more difficult. And the other axis there is it's sort of a structural issue, which is, all three of these countries that seem to have done poorly seem to have autonomous regions within them.
Well, sure, but there's nothing small about China.
Yeah.
And of course, China is autocratic, but it managed to do a far better job of handling the outbreak than the United States. And I certainly am not one who wants to praise many things about China, but you have to give credit where it's due. And it's an enormous country. It has semi-autonomous regions, and yet they were able to bring the virus under control relatively rapidly through pretty intense measures, I must say. Measures that probably wouldn't have worked in the U.S., but some kind of middle ground along the lines of what was used in Europe would or might have worked in the U.S. But the point is, we didn't do anything in the U.S. We still don't have a national policy. So, there's just complete neglect here, and and that's like negligent homicide.
Yeah. So, one way we can. So we'll come back to the policy question again. One way we can get ahead of this is sort of predicting evolution, predicting transmission. Jeff, are there tools now available for us to maybe get ahead of this in the future?
Well, we the best way to predict the future is by seeing what has been happening so far. So we know that pathogens do cross into human populations, and we can see that it happens consistently, and happens in consistent ways. So, for example, many of the zoonotic diseases that we've seen in the past couple of decades have come from wildlife and from the trade in bushmeat and the exotic pet trade and other kinds of trades that deal with wildlife, particularly in the context of deforestation. Deforestation increases human contact and the contact of human livestock with wild populations and whatever infectious diseases they have. So looking at, this is where HIV came from. This is where Ebola comes from. So we can target those populations for surveillance to get ahead of these things in the future. We can enact policies to make those trades less frequent, if we can, and safer for the people who do interact with wildlife populations.
Yeah.
And that's the best way for us to get a handle on it. Like, so for example, the EcoHealth Alliance was collaborating with this Virology Institute in Wuhan. They were doing surveillance of various populations in China for coronaviruses, and they were finding that in populations that interact with wildlife had antibody exposure to various coronaviruses in the range of like 3% or something like this. So they were conducting this kinds of surveillance to know, so we can get a better handle on where the next pathogen epidemic is going to be coming from.
Yeah, Fred, it's sort of embarrassing if you think about in the era of big data and artificial intelligence that, we have been able to really do a good job around this. I know that Johns Hopkins and others have collected a lot of data and putting it out there, sort of an accounting exercise. But, just like we do in other industries, this type of data is going to be quite powerful, right, for us to reach better policies.
Yeah, completely. And I think, that is one of the ridiculous things about, or I find frustrating as a researcher, that a lot of this data about positive cases, case testing, this isn't available. This isn't done by the federal government. These are kind of news organizations like the New York Times or the Atlantic, volunteers and hospitals who are like tabulating stuff and putting it in Excel spreadsheets and sharing it.
Yeah.
At a national level in the US and in other countries as well, arguably, that should be something that should be already being done, right? We should be recording things on a much better, more precise scale. So then when, after the fact, we can go back and analyze. And I would just, make a point that Jeff was making as well. It's no surprise that this, I think for scientists working on coronaviruses, that this has happened. If you read like a lot of the literature, it's okay, it's hindsight's 2020, but there's an awful lot of papers that say one of the next big coronaviruses is going to come from a bat population. It's probably going to be China. And it could be devastating. So it's perhaps surprising for the public and people who don't work in that specific subfield. But I think these kind of things have been known and have been an area of concern for a long time.
Yeah. And also, so once you get the raw materials, there are two issues there. One is having a quick national way to have the raw materials, meaning all the data that we can collect. And then, we have technologies now that could be deployed on that data to create insights. For example, I'm still sort of puzzled by, if you look at the case fatality ratio, across the world, it ranges anywhere from, upwards of 10% to less than 1%. And that's a big range. And so if you ask why is it different, one difference could be that the population infected, you have to control for age, you have to control for pre-existing conditions. But even with those attributes controlled for, you still see a fair amount of variance there. I don't know if you have any insights as to why that is the case.
There are a lot of things that can go into that. We've known for a long time that not only do you have to control for demographics like age and exposure, but the course of the epidemic will affect the number that you calculate. So deaths are typically delayed from positive cases by several weeks or even months. It takes, so as at the beginning of an epidemic, as case numbers are going up, we can count the number of people that we've detected to be infected, but some of those people will later die. So that the number we calculate is all just by who's died and who's been infected so far. We already know that's going to be an undercount when case numbers are going up. And also we know that that number can also be overcounted because depending on how many individuals get infected but don't show symptoms and don't get tested to be positive cases. And that is determined by lots of things, like how strongly are we testing for the virus? What is our surveillance like? What is our reporting situation like? That can vary very strongly over time and across countries and across states and localities as well.
Yeah. And sorry, I was going to jump in and say, and also just on a biological level, we also don't know. So some people seem to be infected and the health outcome is very bad. They get very sick, right? Part of that, I think, we've seen that things have improved over time as treatment plans have gotten better. Like doctors have become, working in emergency rooms around the country dealing with this. There are now better treatment plans in place. So kind of survival rates have gone up marginally. But also we don't know why some individuals are so badly affected. And so it's presumably something to do with their immune system and how their immune system responds. And the immune system, as it turns out, is incredibly complicated. But there are we do have, we actually have a colleague here at the University of Missouri St. Louis, Charlie Clymer, who uses kind of AI approaches and advanced computing to look for patterns in, immune reactions and how that might correspond to your health, your kind of health outcome when you get infected with COVID, to see if we can try to make predictions about what makes some people really sick and how come it doesn't affect others. And that's presumably due to differences, some kind of fundamental difference in how our immune system is responding.
Yeah. So if it is purely a timing difference, we look at, if you look at Italy, Belgium, Hungary, France, they're all higher than 10% case fatality ratio. It might be that we learned how to treat these patients. I think when you go on a ventilator, I think your chance of getting out of that is only about 10%. So that was sort of the initial modality of care, which I think we seem to have moved away from. But there is still significant difference. So what would be interesting also to think about, like you say, Fred, is are there attributes that we can think about in terms of other types of vaccinations people have gone through, other flu attacks that populations have gotten that has some sort of correlation with their ability to resist this. But I haven't seen anything. Yeah,
I think there's some work, and it's so that's the problem, right? So a lot of the kind of communicating new and developing science is tricky because things change very quickly, and that's just part of the scientific process. So some studies that seem promising to begin with turn out to not be as promising as we get more data points. But there's maybe some evidence that if you've recently had an MMR vaccine, there might be some cross-reactivity to, COVID-19, SARS-CoV-2 virus. But who knows? That may or may not be true. It may be that also you might have some cross-reactivity. So your immune system might have been primed because of the previous viruses you've been exposed to. So if you've had previous coronavirus infections, maybe your immune system is better able to respond. But that's speculative at best. And maybe that's going to come out. But it's plausible. We know this happens for other diseases where you have something very similar. You can think of cowpox and smallpox, for example, where cowpox is different from smallpox, but it gives you resistance to smallpox virus.
There's also some evidence with the 1918 flu that might suggest there's an effect in the opposite direction. So 1918 flu was especially harmful, especially deadly to people within a certain age range of like 18 to 35, which is unusual for flu. And there is some evidence that the harmful effects of the 1918 flu on those individuals may have been because when those people in that age range, their first exposure to influenza was a different type of influenza. The first type of influenza you're exposed to usually as a child provides the most immunity. But these were, rather than being the HN type that was the 1918 flu, there was a slightly different one, and it may be that having that initial experience with a different type of influenza primed them to have immune responses that weren't as protective or weren't as good for them, I guess.
Yeah, it's the one thing that we haven't even discussed, and I don't know if we are even able to discuss this, and that is, and we talked about this in a recent podcast, is the long-term effects of people recovering from COVID. For example, the 1918-19 Spanish flu. There is evidence that says about 1 million people who recovered from that later, 10 years later or so, got Parkinson's disease. And so if we see analogs here between Spanish flu and COVID, when you think about the disease burden in totality, it's going to be substantially different from, what people are currently computing, which is like, yeah, you got it, you got over it, and that's that. It may not be, right? And so, the issue here also, Fred, is in a world that is sort of separated, in my view, there is a scientific world, which is increasingly shrinking, and there is a non-scientific world. And the question is, how do you communicate and present information and hypotheses and data in a way that let's call that non-scientific world internalize and act on?
I think it's incredibly difficult, right? I think, perhaps traditionally scientists have been seen as working in some kind of ivory tower and occasionally dispensing some information that might help the population at large. I think that format has to change. I think as scientists, it is really important to try to explain the scientific process, how things work in any kind of medium or format that you can. But I think a lot of it is also just increasing scientific literacy of the general population, making people aware of how the scientific process works, of uncertainty, this idea that we don't always know, but it doesn't mean, just because we're uncertain about some things doesn't mean that other things are, therefore uncertain.
Yeah.
But in this case, I think it's also very difficult because you can see the same data and you can come up with completely different conclusions, right? So, you can take the example, my home country, Sweden, where very early on the government decided, okay, this is terrible, but we're going to not impose these kind of strict lockdowns. We're going to try to actually achieve herd immunity, whereby we infect enough of the population so that the virus can no longer spread. And that's, I think, sensible scientists who are looking at the same data and make a very different policy choice. So I think it is confusing for people. Say, well, how come Sweden's doing it this way and somewhere like the US has ad hoc, sometimes things get locked down, sometimes things don't. So I think this kind of difference in messaging, difference in strategies, even as a scientist, makes it very confusing to know what's actually real and what's not real.
Yeah, it's really challenging. And, talking about uncertainty, Jim, there is, I think there's a lot of optimism around vaccines and drugs. And coming from Pfizer, both of us know what that looks like. So there was a phase 1 trial. I won't mention the company's name. 40, 50 people, typically male under 40, go through a phase 1 trial to look for safety for a product. And 8 of them developed antibodies, and market went up 200%. All sorts of things broke loose. Vaccine, people, setting up manufacturing processes to manufacture this thing. From your perspective, where do you think we are in terms of vaccines and drugs?
Well, we're in very early days, and basically, until we have a drug that's been approved, we don't have a drug. We might have drugs that are in development, but most drugs fail in the development process. They fail for safety reasons. They fail because they don't meet the efficacy requirements of the FDA. They fail for a lot of reasons. They might fail because the company can't make the drug pure enough to meet the FDA requirements, and that can happen with biological agents, and so which these vaccines are. And so having promising early results is great because that's what you want, but it's not very meaningful in terms of actually reaching the market with an approved drug. Of course, it's the first step that you need, but since most drugs fail between that first step and reaching the marketplace, it's nothing to get overly hopeful about. And one thing that you mentioned about the clinical trials mostly being done with men is also something that's a very worrying trend that has been going on for far too long in the pharmaceutical industry. Women are highly underrepresented in many clinical trials, and therefore side effects in women are often not found until drugs are on the marketplace, and that can be a severe problem. So pharmaceutical companies need to be better at recruiting women and minorities, especially, into clinical trials, especially in this case, because minorities are so hard hit by coronavirus and are the biggest victims. They're much overrepresented in the victim group of coronavirus compared to their percent population in the United States, at least.
Yeah.
And so there are all kinds of things that can go wrong, and that whether it's a vaccine or a drug that's being developed. So I don't want to throw a wet blanket over everything. I mean, there are a lot of brilliant scientists who are working on developing drugs and vaccines, and I certainly believe in the process overall. But to say that because these companies are currently at a certain stage means that we will have a drug or a vaccine at a particular date is just magical thinking. We won't have a drug until we have a drug that's been approved, and nobody knows how long that will take and when that will happen.
Yeah, and in the case of vaccines, Jim, I can quite remember. So if you're doing a phase 3 trial in a vaccine, you're doing a placebo-controlled, double-blind, randomized trial. So you have to infect, you have to vaccinate one population and essentially wait, right, to see if it is effective, unless you do some kind of a challenge trial attempting to infect people.
Absolutely. And most vaccines require, or many vaccines require multiple injections over a period of perhaps a year. Or at least 6 months, but often a year. And so you can't even begin to measure whether they're effective or not until the full complement of injections has been given.
Yeah.
And so, we're a long way from giving a year's worth of injections to a large number of volunteers. Now, who knows? Maybe it won't take 3 injections over 6 months or a year for a COVID-19 vaccine, but we'll have to find out how many injections it'll take.
Yeah,
it'll take more than one.
Right. Yeah, that is sort of troubling. So, Jeff, going back to the mutation question, I read that there were kind of 2 types of COVIDs. One is called the S type, and the one other is called the L types. And one of the mutations increased the number of spike proteins on the surface. And if the S and L forms are distinctly different, would that automatically imply we need multiple vaccines?
No, it wouldn't necessarily mean that we need multiple vaccines, because vaccines tend to target specific regions on viruses, and the differences among these types of viruses may not have any effect on what the immune system is seeing. I should also say that some of that evidence about the different types of coronaviruses is still iffy.
Yeah.
I mean, there are lots of viruses mutate all the time. Every time they go through round of production, there's typically new mutations introduced. And what we're worried about is whether those mutations affect the severity of disease or the infectiousness. So it's a little bit early to tell whether those two different types are affecting those things. They could have just been in the right place at the right time, and it happened to be in a place where there was lots of transmission. I think there is some evidence that they're better at attaching to human cells, but how that translates to infectiousness or severity of disease isn't yet known.
Yeah, so I wondered, as Fred was saying before, this transmission severity trade-off, in the big data context, we are picking up a lot of data from all around the world. Are there techniques we can use on that data to not perfectly, but to assign some probability how many different varieties we might be dealing with?
Well, the problem is that there's lots of different sequences of virus sequences out there, and it's a very non-trivial problem to figure out how those sequences affect the biology of the virus. Some of those mutations are basically have no effect. They're in parts of the genome that don't code for proteins or make a change in a protein that is non-consequential. So we're not at the stage where we can look at a sequence and say, okay, this is more infectious, this is less infectious. That's something that people are very much trying to do now. But predicting the important phenotypes, the important viral traits from sequences is still a long ways off.
Yeah. And Fred, so when we think about back to sort of how do we take care of this, one of the policy prescriptions obviously is the testing and tracing policy. We had a lot of difficulties implementing it. There were big differences in quality of tests that seem to be getting better now. But this testing and tracing intervention, unless you do it early in the episode, it is almost impossible to really do it, right?
Yeah, that's one of the really difficult things. And it also has to be like nationwide, or, can't just be, again, there was some early test and trace, but it was quite ad hoc. And now, of course, the virus is probably so widely spread, it's difficult to do that. But even then, look, there will be, and there are, reoccurring local outbreaks. So we're moving past this kind of initial spread, and to some extent, and so we're going to see pockets of outbreaks over the coming months. And so actually test and trace could still be useful, right? Because we might want to know where it's spreading most, where these outbreaks are happening, and to isolate people. I think that's the other thing as well. There's, I have friends of friends who've, gotten coronavirus. Luckily, most of them have been okay. But, you call up your doctor and you ask, should I get tested? The doctor's like, nah, don't worry about it. But you think, well, actually, it would be useful to know if you have the coronavirus, because that has consequences for how you might want to behave or the kind of decisions you want to make. So now the scenario is, if you have any kind of flu-like symptoms, you just have to, assume it's coronavirus and stay home. And for many people, that's not feasible. You'd really, because you have to go into work, for example. But yes, it is disappointing that we had this opportunity to do this and we haven't. And then, as you mentioned, some of the kind of early testing, that's still the testing is getting better, particularly the antibody testing. But a lot of these kind of tests we rely on are PCR based, so we try to amplify bits of the genetic information that the virus carries, so bits of DNA. So we try to see if it's there, and the kind of false positive rates are still surprisingly high. You get amplification when there isn't stuff. You get bad amplification. PCR, although it's a wonderful technology, is still somewhat finicky. And I'd be curious after. Well, I'd be curious to actually know, because I've been involved in helping some local facilities that do some testing, because they're having huge issues with just kind of this fundamental technique, this PCR issues, and so which makes the kind of underlying data quality somewhat suspect, unfortunately.
Yeah, it seems like we are getting better. There was something out of Yale called Saliva Direct that got fast track approval, and I believe University of Illinois developed something, and they're testing all the students on campus or something like that. I don't know the details of it.
Yeah, but there's still a huge variety in like how people are going about testing. So people say, I'm getting tested. And you think, oh, what's it going to be? It's going to be a PCR-based test. Well, even within the PCR-based test, there's lots of different kits and ways that people are doing this. So you're right, it is getting better, but it's taken an awful, I mean, how many months in are we now? And it's only recently, I feel like, have been drastic improvements. So
I should say that the technology may be getting better, but we're not based on the epidemiology that we've been seeing of case numbers, we're not getting ahead of the virus. So one of the ways that we can tell like how well we're testing for the virus is by looking at what fraction of tests come back as positive. So that if we only have enough tests to test the people who are most likely to be infected, we have a very high positive rate. But if we're testing lots of people, then that positive rate goes down, and we're testing all the potential contacts and getting more and more people who aren't negative. And so based on the information that we've been looking at in Illinois, Missouri, and elsewhere in the nation, where our testing efforts are actually falling behind the cases. That the positivity rate, the number of tests per positive rate has been going down since it looks like June here. Yeah. So there's still lots more to do on the testing point if we were to try to implement a test and trace strategy.
Yeah, I have to say, in the tri-state area here, in New York area, at the height of our sort of pandemic here, lack of personal protective equipment, and even simple things that medical professionals need to take care of patients were not available. It looked very much like a developing country in Africa, I have to say. I don't know. I think we have gotten a lot better. Jim, I know that you are involved in, the PPE. It's not just the manufacturing of it, but also, reuse of the PPE as well, right? There are a lot of things that we learned, I think, in this episode.
Right. So sterilization of PPE for reuse is something that can be done by large hospitals that have very expensive hydrogen peroxide-based machines, and a lot of entrepreneurial companies have developed other technologies for sterilizing PPE using UV light, for example, and there are other approaches as well that are out there that can be harnessed, but nothing, I think, has really been produced on a large enough scale to reach the level of every doctor's office and every ambulance and every police station or police car and every first responder, so that they can sterilize PPE as frequently as they need to. And we're starting to see a shortage once again of masks and other sorts of PPE as hospitals, for example, in Texas and Arizona and Florida remain extremely full. And so, and it's impossible for the consumer to buy sterilizing wipes in the grocery store. They're just completely sold out, at least in this area. So, that's a problem. But I think there's another area that is divorced from theories of the disease or testing that is completely being ignored, and that could go a long way towards stopping the spread of the disease. And that is that our building infrastructures are simply inadequate, and they could be fixed. One of the main things that could be done is controlling the humidity in buildings. And there's a study that is by Stephanie Taylor, who's an infection control consultant at Harvard Medical School, that shows that keeping the humidity between 40 and 60% actually makes a huge difference in blocking the spread of the disease in buildings. And there are a number of reasons for this. And one of the reasons is that when the air is too dry, large droplets don't fall to the ground as quickly as they normally would, and they become small droplets, aerosols that float in the air longer. And so they can float to people, and they can also go to surfaces that become contaminated. And also, apparently, airborne viruses like coronavirus aren't as infectious when they float through moist air compared to dry air. And that process isn't well understood, although there are theories to explain it. And finally, our own respiratory immune system works better in greater humidity. And that's been shown through research on mice. And one of the reasons is that our mucus layers are simply more protective in our respiratory systems, and they prevent viruses from attaching. But there are a lot of other reasons as well that all add up to giving us better immunoprotection from viruses in higher humidity. However, but both air conditioning and central heating dry out the air and leave us at the mercy of respiratory or airborne viruses. And nothing is being done in most, with a few exceptions perhaps, in the entire country to address this relatively straightforward problem. And it could be done on a room-by-room basis with humidifiers or building-by-building basis. And that's even ignoring the fact that HEPA filters could be used to filter out the virus, the airborne virus, from building air circulation systems.
Yeah.
So there are specific engineering controls that could be put in place to make buildings safer where people work, and they, I hope they're being put in place, but I certainly know of no examples in places that I'm familiar with where that's being done.
Yeah, it's a one-time investment, right? So you're not just talking about hospitals; you're talking about office buildings, pretty much any building that a group of people are living together, sitting together.
Right.
It's a one-time investment that has a lot of positive effects, not just for COVID, but generally speaking, right?
Absolutely. And hospitals do have this type of, maybe not for humidity, but they do have HEPA filters in place in their air handling. And so, that's a very good thing. And airplanes have HEPA filters. So although the air is recirculated many, many times in an airplane, the viruses are filtered out very effectively. And that makes it relatively safe to travel in an airplane. But any other building that we're in is very unlikely to have those safeguards in place.
Yeah. So in conclusion, I have a question for all of you, and that is, what have we learned from this episode? So if this were to happen again, what will we do? That's question number one. What are the things that we have to focus on? That's question number one. Question number two is, to detect and manage something like this worldwide, it appears to me, at least, we are missing something. We are not really applying technology to the highest level. So if we can identify something beginning to start, then we have a much higher chance of controlling it. So what can we do, sort of a worldwide monitoring perspective, to do something along those lines? So Jim, do you want to go first?
Well, I think the first thing I would say is that the question is not if this happens again, but when it happens again,
Yeah.
because it will happen again. And I think that many of these programs were put in place and were dismantled. So we need to get back to doing what we were doing, and we need to ramp up even those efforts. But I think that one of the most obvious things is that if there is a test available for a virus, then we need to use that test rather than trying to invent our own test in the United States and taking the time that it takes to do that rather than simply implementing an existing test and wasting all of those many weeks that it takes to develop a homegrown test when something is available from the World Health Organization that's perfectly functional and could be implemented immediately.
Right.
So that way, contact tracing and testing could be put in place right away, and we might not have this problem that we have now of people waiting 7 days to 9 days to get their test results, which is still happening as we speak.
Yeah, so rapid development of technology for testing, I think South Korea did that pretty well, and essentially catching it early is the key. But do you also see any sort of worldwide monitoring that might be useful?
Oh, absolutely. But as I say, it was going on and it was shut down.
Okay.
So, we need to go back to that, and we need to be a part of the World Health Organization, and we need to fund the groups that were monitoring emerging diseases in China and in the Amazon and in other areas where deforestation is causing human habitat to encroach on previously uninhabited areas where there are unusual animal life that humans haven't come into contact with before, which are hotspots for disease development in Africa as well.
Yeah. Fred?
Yeah, so, what can we learn from this? Well, I guess one of the things positives, there's not a lot of positives in this tragedy that's unfolding, but some of the kind of epidemiological modeling that's been done over the last few months has been just really like top notch, and the kind of spread of the disease has been kind of accurately predicted and imposed different types of kind of intervention measures. And again, once these things have been imposed, the models have done really well and been able to predict, which I find surprising, to a high degree of accuracy, how these things, how the disease has been spreading. Which I think that's good and that's impressive, and that's why we do this kind of basic science. And I would argue that to continue to do this, we'll need a big investment in basic science. Again, across the board, for that can be in terms of monitoring, but also, some of the most promising vaccines were developed, by universities, and for example, University of Oxford, where they've originally just looking at sort of Ebola, right? So kind of not very much money in this area, but, basic science feeds into industry, and investment in basic science is really important. And during these kind of pandemics, I think it highlights the urgent need for continued and sustained investment, right? Because you never know where the solution to a potential new infectious disease is going to come from. And then finally, I think we just need more scientific literacy amongst policymakers and the general public, because even if we can identify where the pathogen is coming from, how it's going to spread, unless people act on that information, it doesn't matter. And, there's just a huge disconnect between the people who make decisions and the kind of understanding of even basic concepts in science. And so no matter how good the science is or our monitoring is, if people don't respond in the appropriate way, then it's really all for nothing.
Yeah, yeah. It's sort of ironic, Fred, some of these models that even started building in policy knobs on them, which is if you do a stupid policy this way, this is the number.
Yeah.
It's almost like we can't get over it, right? Yeah. It's hopefully we learn from it. Jeff?
Yeah, well, the thing that's been most surprising for me about the way the pandemic has progressed is not that it happened, but how governments have responded and how people have responded. Like, people have been interested in preparing for situations exactly like this since at least 2003, for the first SARS pandemic. Many of the people who are epidemiological models and involved in ecology and evolution of infectious disease, many of those people are my colleagues, and I think it's been surprising to all of us, just like how we have, even it's always hard to know exactly what to do, but we do have really good ideas about how infectious disease works and how interventions work, and even with the best science, governments have decided to do something else. That's what's been most surprising to me, is that we haven't been acting on the best science for one way or another. And and to some extent, that's a policy issue that I'm not as a big policy person, but I wonder, we have organizations built with a built-in certain kind of independence. Like the Federal Reserve is built in a certain way to be resistant to political influence. I don't know as much about that, but all I can think of is wonder, is there some way we could institute through laws where we need to be acting on the best science rather than be at the whims of whoever happens to be in charge at the moment, who sort of dismissing people or hiring people based on how their opinions they're their own.
Yeah, that's a great point, Jeff. CDC and FDA are supposed to be entities like that, but as you say, we don't have explicit laws such as Fed Reserve. Maybe that is something, it's something to consider. I also see sort of a clash between science and popular culture, and that is not something you can correct very easily. I think the latest number I saw was there is a quarter of the country, US, and I think this is generally true worldwide, people believing that the sun goes around a flat earth today. And it is a belief system that science cannot really, I don't know if science can really do a lot about that. And so, so there is a kind of a systemic issue there. I also strongly believe that, as Fred was saying, and I think you were saying, Jeff, that the policy questions are so complex now. It requires experts to make the policy decisions. Just getting a law degree or just getting a business degree might not be enough to make a complex decision in the area of population health, areas like that, life sciences, for example, in general. And so perhaps we need some sort of a competency test for policymakers before they take up the jobs. This problem, I think, is going to get worse and worse as we go into the future. Things are only going to get more complex, not simple, I think.
Yeah, and it's frustrating as a scientist, because you want, as a scientist, our job is to the best of our ability, figure out how the natural world works, right? And we hope to provide information and knowledge and experience so that we can act in ways that are most desirable for us. And having trust in that information, to some extent, means avoiding politics and saying, well, we don't want our political views to influence how the experience we give. And so it's hard even as an individual to like know, how involved should I be in these things? I don't want to contribute to people dismissing scientific evidence because scientists, like everybody, have political opinions. But at the same time, it's impossible to stay totally independent of that, and that's something that is difficult to navigate.
Absolutely, yeah. I think independence, like you say, I think is going to be the key. The systemic effects are going to be more difficult to correct. Hopefully we'll be in a better position next time we go through this. This has been great, Jim, Fred, and Jeff. Thanks so much for spending time with me, and good luck with everything that you're doing.
Thank you. It's been great.
Yeah, thanks, Gill. It's been a really interesting conversation.
Thank you.